All right, and the last pitch will be given by Valentinian, and he will talk about change point detection.
Hi everyone, so today I'm going to talk about the problem of change point detection and segmentation of discrete data using Bayesian context trees. So I'll start the presentation by giving a short motivation and why we're interested in this project. So suppose you have a discrete time series for which you know there is a distribution change, and our focus for this project is to come up with an efficient Bayesian algorithm which can be used to identify the locations of the points for which there is a sharp transition in the distribution or in the statistical generation mechanism. Perhaps one of the most well-known problem related to this statistical problem comes from biology, and it's called the problem of DNA segmentation. And as you might already know, each DNA genome can be transcribed as a long sequence of letters where each observation can take only 4 values, A, C, T, or G. So that's why we have a discrete alphabet in this case. Now, each genome has different segments, and these different segments usually have different patterns, meaning that they have different functionalities. So from a biological point of view, we're interested in identifying those segments because they can help us to better understand the whole structure of the genome. In practice, biologists often model discrete time series using Markov chains. But the problem with that is, with this kind of modeling, is that high-order Markov chains are inflexible and they usually fail to generalize, mainly due to the fact that the number of parameters grows exponentially with the memory length. So this is why, throughout this project, we try to use only parsimonious models. And we're going to use the so-called variable memory Markov chains, or in short, the VMMC models, which is which is basically a sparse ordering Markov chain. So let's see how it works. Suppose you have a discrete alphabet, a binary discrete alphabet, which basically contains only 2 elements, a 0 and 1. So each observation can be either 0 or 1. And let's consider a third-order VMMC. So there are 2 important things to note about VMMC. First of all, each VMMC can be graphically represented as a label tree like the one we have there. So that's a third-order variable memory Markov chain. And what you can see is that for each specific leaf, we have a specific probability vector. So this leaf has this leaf has parameter theta, probability vector theta 1. This one has theta 0, 0. And the second thing to note about VMMCs is that the memory is a deterministic function of the past observed values. So what does it mean? So for example, if you want to find the probability that your current sample xt equals 1, given your last observed sample was, for example, 1, then graphically it means you have to take this path here up until you reach this node, this leaf, which has distribution theta 1. So in order to find p of xt equals 1, you just have to select theta 1 of 1. Another example might be this one here, where you want to find the probability that xt equals 0, given your past 2 observed samples were 0, 0. So in that case, graphically, it means that means you have to take this path up until you reach this leaf, for which again you know the distribution. So in this case, the memory is 2. So the first problem I want to address in this talk is how do you, so you're given a discrete time series. The question is, how do you find the VMMC which best fits your observed data? So you can use the so-called Bayesian context tree framework. And before I go over it, I just want to introduce some notation. So we have our finite alphabet A, which let's say contains m elements. We have the maximum memory D. We denote by curly T of D the collection of all proper tree models T on A with depth no greater than D. Each leaf is going to be referred to as a context. We're going to store for each leaf, we're going to have a probability vector theta s, which now has m elements because we are operating within an m-array alphabet, and the observations are going to be denoted by x. Okay, so the first thing to do is to put a prior on all the three structures that lie in your collection curly T of D. And we've chosen to work with this kind of prior, which penalizes through this term the larger and more complex trees. So we have alpha here, which is a hyperparameter between 0 and 1. And we have absolute value of t minus 1, where absolute value of t is the number of leaves in your tree. So basically what we're doing, so the more leaves you have, the more the larger your model is going to be and more complex. And in order to avoid overfitting, we're penalizing that by an exponential amount. Then for each leaf in our tree, we place Dirichlet half-half prior. So this is standard in Bayesian analysis. And then using Bayes rule, you can show that the form of the posterior looks something like that, where this term here, PD star, is called the evidence or the prior predictive likelihood of the data. And it can be calculated by summing over all the three structures in your curly T of D. So like in any Bayesian approach, this term is quite hard, is intractable to calculate even for low values of D, because actually theoretically you can prove that curly T of D grows doubly exponentially in D. So this is quite common in Bayesian statistics. But what we can do actually is to use the so-called context tree weighting algorithm, or in short the CTW algorithm, which allows you to calculate exactly the prior predictive likelihood, because we used those kind of priors that I talked about before. So this is really good. And then there's another algorithm called the BCT algorithm, which can help you to find the map tree model. Okay, so let's see, given that we have some tools now, let's see how we can apply them to the problem of change point detection, where in this case the observed sequence is not stationary anymore. And just to remind you, our goal is to infer the number of change points and their locations in the same time. So basically what we're going to do is to observe the entire sequence, partition it into piecewise stationary segments, and then for each segment, we're going to model each segment as a homogeneous VMMC. Some further notation. So we have L, the number of change points, and we're going to store the change point locations in our vector P. So the idea is quite simple. From position 1 up to position P1 minus 1 is going to be the second segment. From P1 up to P2 minus 1 is going to be the second segment, and so on. Again, we define the priors. So we have a flat uninformative prior on the number of change points, where L max is the maximum allowed number of changes. Then we've chosen to work with a prior which penalizes shorter segments. So basically, if you have a look at the form of this prior, we're basically multiplying the lengths of the segments. And basically, this, as I said earlier, this prior penalizes shorter segments and avoids obvious overfitting. So we're not letting two change points to be near each other. And then, as expected, for each, because each segment is modeled as a VMMC, we place the BCT priors that I just described. So the posterior, again, using Bayes rule, you can show that the posterior is proportional to those three terms here, where P is basically the sort of the prior predictive likelihood of the entire sequence times the two priors that I just described. And the nice thing about this approach is that actually, because we placed independent priors on the different segments, basically you can show that this quantity here is just the product of the PDs of the segments. And as you remember, we could calculate the PDs using the CTW algorithm exactly. So in other words, we can have effective inference on P and L, meaning that we can sample directly from this posterior distribution without needing to sample or even estimate the models within its segment. So this is really powerful, and this would make our Bayesian approach really fast. Okay, so I'll finish up my presentation by showing you one experiment that we've done. So here we have the bacteriophage lambda virus, which is usually used as a benchmark data set for comparing different algorithms. So it's quite long. It has around 50,000 letters. And we run our Metropolis Hastings MCMC sampler for around 700 iterations, so that's not that much, with D equals 10 and L max equals 10. And these are the results that we got. So on the left here, you have the posterior over the number of change points. And what you can already see is that we have, most of the mass is placed for 4 changes. And you can see their location on the right-hand side graph. So basically, those are the 4 most probable change points. And we have 2 other change points which are more likely, one located here and one located here. And in terms of results, we also compared our map estimates to those obtained by Braun and Muller in their 1998 paper, which is quite old, but it's still used as a benchmarking paper. And actually, we've also compared our results to the true biological positions, locations which were identified in the lab using different biological tools. And what you can see is that our estimates are in fact closer to the true value. And what's really nice is that if you zoom in around those points, you're going to see that the posterior ranges actually include both the true locations and the locations obtained by Braun and Muller. So that's pretty much all I had to say. The only thing I think, the key point, the point of this talk was to show you that we can do effective Bayesian inference, which can be really fast, mainly because we can sample directly from the posterior distribution without the need to estimate or sample the models within each segment. So yeah, that was all. Thank you.
Thank you, Valentinian. Any questions from the audience? Oh, Lenny.
Can you apply this in a data streaming setting? Is there some kind of online version of the algorithm that you can leverage similar computational gains?
Yeah, probably you can. I think there are some algorithms which work exactly for the online thing. We haven't thought about it, but ideally you'd have to have a discrete alphabet so far, as far as I know. So as long as you have a discrete alphabet, it should work, because basically what you have to do is to compute the CTW every time. So I guess it can be done to an online setting as well.
Okay, thank you.
How important is this memory property that you have? Like, what if I limit the memory window, or I don't have any memory at all? How much does this change?
So basically, at some point, I also said that you can find the map3 model for each segment. So we ran also the BCT algorithm, and we found that actually the map3 model has, in most of the segments, a length around, let's say, 3 or 4, maybe 5. So running with 10, it's more than enough. In terms of results, as long as you're above that 5, I guess you're all right. So it's not going to make any difference.
But computations are much quicker if you don't have a memory window or a smaller window.
So for example, this CTW algorithm works in, the order is, I think, NMD. So it's linear in both the number of samples, the number of elements you have in your data, in your alphabet, and the depth. Oh. The only key problem is that, in the way I coded it, you have to construct a very large tree, and usually that's computationally inefficient. But to really run the algorithm is really fast. So.
Okay, so then thank you, Valentinian, and thank you to everyone in the elevator pitch. Okay, I think we have a break until 15:15 with coffee and 30:30. Okay, great, until 30:30.